Thank you very much. I'm Brian Freed. I'm the founder and chief scientific officer of RheumaGen. In addition, I'm the professor of medicine and immunology at the University of Colorado Anschutz Medical Campus, where I run the Center for Clinical Immunology. The technology that I'm talking about this afternoon was developed there and is licensed to RheumaGen, but there's a partnership between this, which I think we've heard at this meeting is very important. So a partnership between RheumaGen and the University of Colorado. We're interested in several autoimmune diseases. I'm really only going to talk about refractory RA today. This is obviously a serious problem. Depending on who you talk to, you may hear that rheumatoid arthritis is well controlled with current biologics. But the truth of the matter is, as you can see here, up to 50% of the patients are unable to work even on these biologics. Most of the patients, 3/4 of them, are dissatisfied with their treatment. But more importantly, a hard figure of at least 10% of the patients who are not controlled at all. They're refractory to the disease. For example, 10% of the RA patients develop interstitial lung disease, so really a fatal manifestation of RA. So the disease is clearly not well controlled. Our target for this is the HLA gene. Many of you, I think, are familiar with this. The HLA molecules normally they present viral peptides. We're interested in one locus here today, DRB1, that will present viral peptides, and that's how you trigger an immune response, for example, to COVID and other infectious agents. But sometimes it goes awry, and it presents self-peptides, such as collagen or citrullonated vimentin, and triggers rheumatoid arthritis. In the case of multiple sclerosis, it may be presenting myelin basic protein and again triggering an autoimmune response. Now, one of the things that we've been able to do is map this specifically. And so one of the things that we've identified is the fact that the strength of the peptide binding to an HLA molecule is critical. Now, as I pointed out in the previous slide, there's many different alleles. There's over 2,000 alleles. So only a small percentage of the people are going to have the alleles that are susceptible to it. And the reason for this is because the T-cell receptor can only recognize a peptide, such as collagen, in the context of an HLA molecule. So it has to bind that molecule in order for this to work, in order for it to trigger. And this figure here shows that in the pink on the far right-hand side, if the strength of this signal is very, very strong, that leads to an autoimmune disease, that leads to organ allograft rejection, that leads to destruction of virally infected cells. If it's in the yellow territory, a weaker signal can be more associated with allergies, B-cell responses. If it's in the green range, again, somewhat weaker, that's where you get into tolerogenic signals. Well, what we're trying to do here is to actually turn the pink signal, the very, very strong signal, into a no response by editing the HLA molecule in such that it can no longer bind that peptide. So if it can't bind a peptide, T cells can't recognize it, there is no immune response. So this slide shows in the figure in the center where you see resistance at the top and susceptible at the bottom. These are the different numbers of alleles, as you can see here. And at the very bottom, the most susceptible one, for example, DRB1 0401, highly susceptible, and it also binds a lot of collagen. So that's what those peaks are showing you. The peak shifting to the right means collagen binding. If you look up at the top at a resistant allele, it simply doesn't bind collagen at all. And therefore, it's not susceptible. So really, there's a genetic susceptibility that's triggering this. And we can see in the center line by the peptide repertoire that the 0401 allele has a strong preference for acidic amino acids in the middle of the molecule where it adheres, whereas the resistant allele prefers basic amino acids in that site. So since collagen has an acidic amino acid, it naturally binds very well to 0401, but can't bind to 0801. So we might want to use this, but the difference between the susceptible and a resistant are 13 amino acids. We can't change that many amino acids without it triggering a rejection of the cells that we edit. But we can map it down to a single amino acid, and that's in fact what we've done, and that's where our first patent covers it. So when we change just the one amino acid, shown here on the left-hand side, from a blue, a basic amino acid, to a red amino acid, acidic. So we insert an acidic amino acid in that site. It can no longer bind collagen, but the body doesn't recognize it as being foreign. It's too small of a change, so it doesn't get rejected. So that means we could take a patient's bone marrow out, edit one amino acid, basically plastic surgery on the immune system, and now it no longer binds the peptide. So that's the approach that we're talking about here, but we actually have now 4 other issued patents, and I'll just mention them briefly. The second one is this occlusion approach, where we can edit the major pocket that holds the peptides intact. And when we edit that, we can basically fill it up. Think of it as like a billiard table where you stuff paper towels into the pocket. By putting a larger amino acid in there, we plug it up, and now peptides that have a large aromatic amino acid can no longer bind. And then the third one, the anchor editing, is if we block this site, it's common to all HLA molecules. If we block this site, they will no longer bind any peptide. So we have a very highly specific approach, one that in the first case. The second one, the occlusion method, can block perhaps 50% of the peptides that bind. And the third one blocks all of the peptide binding, but only to the allele that we edit. So you still have other HLA molecules that are capable of responding and presenting. So you're not globally immunosuppressed by this model. So when we do this, if we do a bone marrow transplant where the HLA molecule has been edited, the cells that come out of the bone marrow are macrophages, and they're triggering the immune response. Macrophages only live about a week, so within a week you have new macrophages coming out of your bone marrow that no longer can trigger the autoimmune response. So even though you have collagen-reactive T cells floating around in your body, they need to be re-stimulated about once every 10 days, and the new macrophages can't stimulate them. So our therapy, RG0401, the RheumaGen therapy, is working at the very earliest signaling event. All of these other inhibitors act later. They let the T cell get activated and then try to stop its function. The problem with that is twofold. One is you've already let the collagen-reactive T cells get going. And secondly, it's much more global in its immunosuppression. They're suppressing all kinds of T cell functions, not just the collagen-reactive ones. So this is what the therapy looks like. And by the way, this is therapies that we do all the time at our Center for Clinical Immunology. So we do a GCSF mobilization. That's how we mobilize bone marrow stem cells for all bone marrow transplant patients. Then we collect them by apheresis and isolate the CD34 bone marrow stem cell. And then we do a Cas9 gene editing approach. Now this CRISPR approach that we're taking here, we cut the gene open, and instead of just replacing the one nucleotide, because we really only have to change one nucleotide, one amino acid, we actually insert a whole new gene in there with a marker that allows us to track it and isolate the edited cells. Then the edited cells are collected, frozen down, and can be transplanted really at any time. The beauty of this therapy is that RA patients are not heme malignancy, so there's no critical time factor. They can be treated 6 months later at any time that's convenient. And secondly, because it's not fully myeloablative, the therapy to put them back in, all we need to do is make space in the bone marrow. So this can be done as an outpatient therapy. There's over 600 centers in the United States where you can get an outpatient infusion. So it's really not limited to a bone marrow transplant unit. Again, the patients can be infused with these frozen cells. It's a one-time treatment that's curative because we replace the HLA molecule with one that can no longer trigger the disease. And finally, it's not lymphoablative. So we're not really knocking out all of the patient's T and B cells, which is common with many autoimmune diseases. The problem when you knock out a patient's T cells or B cells is you make them very susceptible to infection. We don't have to do that. All we have to do is replace the bone marrow stem cells, and we can leave everything else intact. So RheumaGen's pricing model, the way that we have this collaborative effort, it really reduces the cost of getting the therapy to market by about 90%. We've done this before with a mesenchymal stem cell product used for diabetic foot ulcers, so we know that we can actually achieve this. But as you can see here, there's several factors going on. So one is it's less expensive to get this therapy to the patient. Secondly, there's a large market. I mean, this is, obviously 1% of the population suffers from rheumatoid arthritis. So it's a very large market that we're dealing with. We're really looking at the cell therapy range here, more in the line of a CAR T cell. So about a $400,000 treatment, not $2 or $3 million, which I think is very important for the payback via Medicare. So I've only mentioned really the rheumatoid arthritis and the one therapy at the top, the RG0401, but we also have preclinical data on several other therapies such as multiple sclerosis and type 1 diabetes. In both of these instances, we're able to edit the HLA molecule that's associated with that disease and block its ability to trigger a T cell response. So those are the three that are currently moving along the fastest. Obviously, we're starting with rheumatoid arthritis, which is a chronic disease and sort of very amenable to therapy, as is multiple sclerosis. Obviously, with something like type 1 diabetes, that therapy has to be initiated within weeks after the patient gets first diagnosis. So that's a trickier disease to treat. So that's why we're developing this therapy in RA and MS first. We have a very experienced group of advisors. George Sett, who's well known as a CAR T cell in autoimmune disease. He's a rheumatologist and an advisor for us with really perhaps the best experience in the world in terms of cell therapies. And then our other group of we have Shani Dwyer in Australia, as a former head of the regulatory agency there, and Bruce Goodwin, who was with Johnson & Johnson for many years, is again with our rheumatology group in Australia. And then I'd like to finish just with a quote from our CEO and co-founder, who just happens to share the same last name with me. But really what our focus here is not on palliative care, but it's really on curative. And all of these therapies are designed such that they'll stop the disease and completely block it. I just have some contact information. There's also more data online. We just recently published all of the basic science data behind it. I didn't show you a lot of that today, but it's just been recently published in Arthritis and Rheumatology, showing that you can edit the molecule. We have mouse models that are completely resistant to collagen sensitization. And so again, that's all published online, but here's our contact information. If you'd like to get in touch with us, we'd be happy to talk to you. We've just finished our Series A round. We raised $15 million in the Series A round. We've raised $20 million overall, and we're well positioned to start the clinical trials. Should be in the fourth quarter of 2026. So thank you very much.